So good afternoon, everybody. I'm very happy to present you this afternoon our work about 3D and spatial imaging to understand tumor-associated macrophages role in glioblastoma. I'm Aurélie Tchoghandjian, and I co-lead the gliom team in Marseille with a neuro-oncologist, and our team is pluridisciplinary, and we have a bench-to-bed and a bed-to-bench approach. So glioblastoma is the most frequent and aggressive brain tumor in adults. It's really a devastating tumor with a very low median of survival. So these tumors could be very huge, they are necrotic and highly angiogenic. And the standard of care is a surgery following by chemotherapy and radiotherapy, so temozolomide-based chemotherapy, concomitantly with radiotherapy, and then chemotherapy alone. And despite this treatment, tumors always relapse. So why it's so difficult and complicated to treat these tumors? And one part of the answer resides in the cellular heterogeneity of these tumors, and they are composed of several types of cells: astrocytes, lymphocytes, neutrocytes, dendritic cells, macrophages, and microglia cells. And all these cells have a tumor supporting role. And among these cells of the microenvironment, the microglia and the macrophages, so the TAM population, is the most abundant one. And so if we have a closer look to this TAMs, so there is this microglia and these monocyte cells, and during early state of tumor growth, the microglia cells are the most abundant cells, and then there is also high recruitment of the monocytes in the tumor. And these TAMs are highly important for tumor progression, immunosuppression, and cerebral edema. So they really play a key role in this process. So what are exactly these TAMs? So as I told you, in the brain there is, sorry, I just take the pointer. So in the brain there is immune-resident population, so these microglia cells, which are derived from the Yolk sac, and we can find also monocytes during tumor growth. So these microglia cells are found in the parenchyma, and they invade the tumor at early stages. And this microglia, this homeostatic microglia, is highly branched, and when they enter in the tumor, the cells become amoeboid. And another TAM population, so is derived from the bone marrow, so from the bloodstream. And so here you can see all these green dots running in the bloodstream, and these cells arrive in the tumor. And so when these monocytes arrive in the tumor, they can differentiate in monocyte-derived cells, these P2, P3 cells, or in monocyte-derived macrophages, these P4 and P5 cells. And they have a tumor supporting role, and these P4, P5 are like these M2 macrophages. So how to reprogram the TAMs to restore a functional immunity? And to do so, in the lab, we use Smac mimetic. So at first, Smac mimetic is a pro-apoptotic molecule. It's an antagonist or inhibitor of apoptosis proteins. And what we did in the past and what we showed is we used these molecules to better understand the YAPs in the tumoral cells. And so we treated the stem cells derived from the tumor, and we showed that upon this treatment, we can differentiate the cells upon astrocyte-like cells, so GFAP positive. So in anorexia, we increase GFAP. And in hypoxia, because glioblastoma are highly necrotic and highly hypoxic tumors, what we showed, we did not induce differentiation of the stem cells, but rather cell death and apoptosis. And this treatment also decreased tumor growth and increased mice survival. So what we know also about this molecule is that the inhibitor of apoptosis proteins are involved in FKB and TNF-alpha signalings. So in addition to have this role on the tumor cells, they probably also have a role in the modulation of glioblastoma signaling. Glioblastoma inflammation. So what is the role on TAMs remodeling? Because these TAMs are the most important immune cells in glioblastoma. And this project was a PhD project of Emmanuel Snazelfazi, and to investigate this, Emmanuel developed light sheet imaging and performed clearing of mouse brain. So we orthotopically injected tumor cells in the brain, and then after tumor growth, we take out the brains, and then we perform the Woolman stainings to analyze the vessels here in magenta, the tumor cells in red, and the immune cells in yellow. And by using this technique, we could investigate the spatial distribution of the immune cells by using this technique, because in glioblastoma, as in most of the solid tumors, these immune cells are located outside of the tumors. And to analyze, we used IMARIF software, and we performed segmentation of the tumor cells, and then segmentation of the blood vessels, and also of the immune cells. And then we quantified the density of the immune cells present only in the tumor to understand if the treatment, the Smac mimetic treatment, could modify the density of the immune cells inside the tumor. So this is the results what we obtained. So to do this analysis, we used really a macroscopic images to have the bulk of the tumor, and then we could reconstitute the bulk of the tumor. And so on the upper images, we have the control conditions, and the SMG, so the Smac mimetic condition, we have the tumors after treatment, 2 weeks, 3 weeks, or 4 weeks after treatment. And what we can see is already after 2 weeks of treatment, we can see an increase in the amount of immune cells inside the tumor, which is, let's say, quite stable during tumor growth. So the inhibition of YAP protein increased the density of immune cells inside tumors. Then we wanted to know if, in addition to this increase in density, we have an increase in infiltration. And we used the same brain, but we used single cell resolution to better analyze the immune cells inside the tumors. And we split the tumors in 4 layers. In violet, you have the immune cells located inside the tumors, and in yellow and white, the immune cells located at the periphery of the tumor. So by using this hierarchical clustering, we could quantify the repartition and the distribution of the immune cells inside the tumors related to the border of the tumors. And what we obtain is that with the treatment, we can find immune cells inside the tumors, but much earlier, and already after 2 weeks, we can have a lot of immune cells in the center of the tumor, and this tendency is maintained during tumor growth. So we have promotion of immune cell infiltration inside tumors when inhibitors of apoptosis proteins are inhibited. So we know that immune cells can also come from vessels. They can use vessels as a route to infiltrate tumors. So we also wanted to know what is the distance between the immune cells and the vessels to understand if we can find more immune cells far away from the vessels, meaning that the infiltration is also promoted. And here again, we segmented and we performed a hierarchical clustering based on the distance of the immune cells related to the vessels. So when the immune cells are close to the vessels, they are in blue, and when they are far, they are in red. And so this analysis revealed that upon the treatment, we have immune cells located at distance of the vessels. So we promote here again infiltration of the immune cells with the treatment. So to conclude this first part about the spatial location, so our treatment, in addition to have an effect on the tumor cells, we could promote the density of the immune cells inside the tumors, but also the infiltration. So then we wanted to determine which immune cells could be involved in this process. And so we used a mass cytometry to phenotype the different immune populations. And we used this hierarchical clustering, which subdivided the immune cells into 16 populations. And here are the results. So we analyzed this after 21 or 28 days post-graft. And what we could see is that with the treatment, actually we increased the active microglia, we increased the CD8 subtypes, we decreased the anti-inflammatory macrophages, so these P4, P5 macrophages, and also we increased the monocyte-derived cells. So these cells, which are increased in density and in infiltration in glioblastoma cells, so they are not anti-inflammatory macrophages. This population is decreased, so these bad and anti and pro-tumoral macrophages. We increased CD8 T cells, and we increased CD8 T cells, and we also increased this activated microglia. So as microglia are the brain-resident immune cells, we wanted to know a little bit more about these cells in our model. And so here are the different states of microglia that we could find in the brain, as I presented you at the beginning. So homeostatic microglia is highly branched, and then when they enter in the tumor, they become amoeboid, and they could be activated, and they change again the morphology. So to identify macroglia, we used TMEN 119 marker, because there is no marker of there is no specific marker of activated macroglia, because these activated macroglia share common marker with the monocyte-derived cells. So by using only one marker, this was not possible. And to highlight this macroglia activation, we performed co-staining with MHC2. So we used again light sheet imaging to have really microglia, the entire organ with the microglia, and really to catch the morphology of these microglia cells. And what we obtained is like in a control tumor, we could have several quantity of TMEM cells, so reflecting that in these control tumors, microglia could be at different state of activation. And when we treated the mice with Smac mimetic, the shape of the microglia cells changed. They look like activated microglia, and the TMEM staining really decreased, so suggesting again this activation, this promotion of activated microglia cells. And we quantified again the location of microglia related to vessels, and here again we could analyze that the microglia cells are found deeper in the tumor with treatment. So then we also wanted to know if there could be a crosstalk with the CD8 T cells, because the mass cytometry results highlighted that we have an increase in the CD8 subset. And so we performed enrollment of both of TMEM positive cells and CD8 T cells at 2 different times after treatment. And what we could observe is, so in addition with the increase here again in CD8 positive cells, so we confirm this increase here, we got a decrease between microglia cells and CD8 T cells. So we decrease this contact between these two types of cells. So meaning that this TMEM microglia is probably have inhibitory effect on this CD8 T cells. So I did not show you here these results, but we showed that this reactive microglia upon the treatment is pro-inflammatory and has anti-tumoral properties. So to conclude this part of immune cell composition after our treatment, so in addition to a decrease in anti-inflammatory macrophages, an increase in CD8 T cells, so we have this basal microglia, which are present in clusters in control tumors, and which are becoming activated in pro-tumoral and inflammatory cells and anti-tumoral cells upon our treatment. Then we wanted to know which inhibitor of apoptosis could be involved in this phenomenon, because our treatment, so this Smac mimetic, could antagonize 4 different inhibitor of apoptosis proteins. So thanks to our collaborators in Freiburg, we had access to single-cell databases coming from human glioblastoma, and we spotted out the expression of our inhibitor of apoptosis protein, and we found that MLIP had an expression specific of these tumor-associated macrophages, so of these monocytes and of this TAMs population. So all the TAMs, actually the bone marrow derived or the microglia cells. So this results that we obtained is related to the inhibition of MLIP protein in our model. And also we analyzed the expression of MLIP in tumor cells. And in glioblastoma, the 4 different tumor subtypes have been identified: the astrocytic-like, the MES-like, OPC-like, and NPC-like by single-cell results. So, and what we find is like we correlated, so the MLIP expression with the MES-like subtype. So this subtype is highly plastic and is the most aggressive subtype in glioblastoma. So this result was highly interesting. That's why we want now to understand the role of MLIP in glioblastoma. And in the team, we started to perform some experiments to understand its role. And we generated cells downregulated MLIP. So we used the glioma cell line, and we downregulate MLIP expression, and we grafted in the brain these cells. So the cells expressing MLIP and the ones which did not express MLIP. And we treated or not these mice with Smac mimetic. And what we got, so we got an increase in mouse survival with the treatment. But when we graft the MLIP downregulated cells, we increased significantly the survival of the mice, and the treatment in that condition could not really promote increase the survival of the mice. So meaning that when we remove the target, the treatment has no effect anymore. So this result was very interesting for us, and now so we want to go deeper and to analyze how MLIP expression could modulate the microenvironment, if there is something here again. And we performed again CD45 Wallmont staining, and we injected the cells, the control cells or the cells downregulating MLIP, and we treated here again the mice or not. And in our control condition, we could increase again the quantity and the density of the immune cells. And really, in a very interesting results, what we obtained is in the cells which downregulate MLIP, we did not increase, in that case, also the immune cells, but we decreased this amount of cells, and we decreased it even more with the treatment. So to understand what is going on, because probably other cells are involved, maybe other cells of the microenvironment are involved. So to have like a big picture, we started to perform maxima, so spatial proteomics, and we set up a panel of 57 antibodies. We mainly use antibodies from Miltenyi Biotech. We use 3 other antibodies, and we used only conditions that are recommended by Miltenyi Biotech. And so this is the panel of antibodies. So it took some time to set up the experiments, because we had to test several concentrations to increase concentrations when the antibodies did not work at first. And finally, most of the antibody worked. So it was a very good result. So these are the preliminary results, the results of the test to validate the antibodies. And here I present you results about the tumor cells. So we used several markers to identify the tumor cells, the proliferating cells, the, the stem cells, the hypoxia. And we did, and here is the merge actually, so we can see that some red tumor cells invade the parenchyma, and that most of the tumor cells can proliferate, but also we have some blue clusters which are not immune cells. So this is infiltration probably of cells of the microenvironment. So we did also a panel to analyze vessels, angiogenic vessels, or to stain adhesion molecules. Here is a merge. And we did the same for immune cells and macrophages. So TMEM to analyze the microglia cells. So we can see here again the clusters of microglia and the homeostatic microglia in the parenchyma. And we could see that here, for example, this microglia is probably activated because it co-expressed MHC2. So, and this is a merge. And this is the same for the macrophages, blood-derived macrophages. Here is a merge. And also we did a panel for lymphocytes. So in yellow, it's CD45. And for lymphocytes, this macroscopic view is not working very well because there are very few cells in glioblastoma. So we have to increase the zoom to see these lymphocytes, but we can see the CD3 lymphocytes, CD8 and CD4. And we also have a panel for neural cells, so to analyze oligodendrocytes, astrocytes, neurons. So this technique is working quite well, and so now we are preparing the samples. So samples with tumor which express or not MLIP protein. And we will analyze the tumors and the microenvironment at different time of the tumor growth. So in conclusion, 3D imaging is a powerful method which gives results that cannot be generated with another technique. And the maxima is promising, most of the antibodies worked. And so far we did not start to build a strategy of analysis. And this is actually ongoing. And what we could see is that YAP inhibition and MLIP inhibition promoted microglia reactivity, infiltration, and anti-tumoral function. And these techniques will now allow us to unravel the role of MLIP in glioblastoma progression. So thank you for your attention, and I would like to thank all the team, all our collaborators, and Miltenyi Biotech for your invitation, and also ARTC Sud. So this is the patients association with which we are working with. So thank you.